Grade 11, choice 2. Second lesson.
26th of June.
26th of June.
Action.
Why so detailed?
Okay. So the first thing was you had to shade. And there were two patches that were shaded. The one was up here, and the other one was down here. The rest of it wasn't shaded, and you had to draw this little region across the middle where it wasn't shaded. Then the second thing it said to you, indicate the color that the reagent would have shown. So iodine solution turns blue-black. Nothing else. Blue-black. It's the only thing you're allowed to say. Blue-black. So that would have been in the regions where you shaded. And it's yellow-brown or red-brown or reddish-brown or yellow-reddish-orangey brown. I don't care what you say. In the unshaded regions. Yep.
Ma'am, I think that I only got 1 mark for either iodine solution or blue-black, because I wrote them both.
Okay, I'll look.
Thank you.
Okay, then question 4.5, why do they decide to test for starch? Because the excess glucose is converted to starch and stored. Now, starch contains glucose. I don't know what you mean. Okay. Then there was a question about what process would have occurred in the cells of the leaf and produced this carbon dioxide that is under the cardboard. It's cell respiration or cellular respiration or tissue respiration. I don't care what you call it as long as it's one of those three. I'll get back to this anaerobic respiration that some of you wrote and explain to you why this was a problem. 4.6.2, where does it occur? Which organelle? Mitochondria. When does it occur? So it says photosynthesis requires light to occur. When does the process that you named there occur? 24 hours a day. Or during the hours of light and dark. Okay, you can't, it's not accurate to say during the day, because do you mean a 24-hour day or do you mean the time of the day when it's light? Okay, so some of you here in 4.6.1 wrote anaerobic respiration. Yes, it does. But then you can't write mitochondria here, because anaerobic respiration is only the first part of respiration, glycolysis. That occurs in the cytoplasm, it doesn't occur in the mitochondria. So you had to lose 1 mark there somewhere. Okay, check your totals there, guys. Cyrilla, please bring yours. I might well have, it's a well-known fact, I'm not like 4 eggs versus 6 eggs. You know I don't do it. Sydney!
Yes, ma'am.
You wanted me to check something?
I'm counting those.
No. Slow little cookie poop.
I was doing it, you asked me to do.
I know.
Ma'am, do we
But I
have to put
expected
these in
you to
our
do
file?
this.
Yeah.
No,
iodine
shh.
solution.
No,
Ma'am, I'm just showing you where it was.
Okay. So, okay. You got that mark for the blue-black.
Oh, yeah.
You got the mark for the shaded. You got the mark for the unshaded. What you didn't do was put the color with the unshaded region as well with iodine solution. That's what you
No.
Okay, does that make sense?
Yeah. Ma'am, must we put these in our matric portfolio files?
Matric portfolio files.
Yes.
Some of you maybe not.
And so the answer was A. And here it is ethanol, and it states in plants. But doesn't it occur in...
Wait.
How did you get 2 marks?
Because
Right.
was.
It wasn't. Oh, no. So you put L. You put 2. No. No, listen to me. Not doing respiration. Because used to photosynthesis, it's not used to respiration. It's actually broken down during respiration.
Nice one. Got 88.
Does that make sense?
Yes,
I'm sorry.
1% of that.
Actually 2%.
No, it's 78.
Oh, I see what you're saying. They should be marked and the other mark is yellow, right? Okay, right. Is everything else correct?
Yeah.
Okay. Wait, you don't go? No.
I don't know what you're expecting to say.
Did I
Missed out 2 marks.
Okay, that's fine. Can I, no, listen. No, I'm quite right. And I'm really good with figures. Don't touch every tip. Okay, so it's the one reason why I don't write the sign because I can't add. So if I just put the tips on the top. No, shh. I think it is those.
Ma'am, can we punch our papers?
Yeah. So there's a punch over there. Just don't move anything on the punch. Look. What is that raise in terms of percent? Yay. So it helps you have your different bathroom to the one she went. I don't know. Social distancing and all. Tony, you are too close to Jade. Step away from the Jade.
Jane, what did you get for 3.2?
A for assessments. I'm sure that's not what A is for. I have to be honest, I don't care. This stupid file system here, I don't know.
Why do we do it?
In the trick, I can understand it, although it's also not an ideal system in the trick. In the trick.
Yeah, just I'm not looking at the rock.
It's okay, it looked like that beforehand. Are you rocking on my chest?
Then my mother would hit me with a rock. I didn't read the question. And ever since grade 4, she said, read the stupid question.
Ask your mom if I can hit you with the rock as well.
Ma'am, you don't have a responsibility yet. Please do.
Awesome.
Thank you.
Then can you use your body parts for dissection?
Ma'am, didn't I have more worksheets in this that I handed to you that I don't think I'll put in the portfolio?
I don't get paid to put your stuff in the book.
No, but you said please hand it in because I'm a mark or so extra.
Lost it.
Now, what about our supplements?
So there's an embargo on results. Let me read this to you.
No, 2 things. What do? You have 4 things
it means the following. I have to show you this because it's the stupidest thing I've ever seen. With the what?
The baby stuff.
Yes.
Still old, man.
Are we really done?
Steve, why? Make it work. You laugh at me. Just say make it work. Put your mask over your nose. Okay. No, I'm fine with you doing that. Well, put it over your nose and then put the stick out through the bottom.
Ma'am, do you know how complicated that's going to be? That's going to be like this. Like, how do you even...
It's on me, so don't say how would I even. It's how would she even. Okay, why is this not going on?
Ma'am, why do you always have to change your shoes and wash your shoes? Not you.
Because the droplets fall down.
Because I got into bed yesterday with my shoes on and my mom showered. I mean, I don't shower at night.
Good.
I wore my face every day of school. No, I know it was
No, everyone has 4 things in their portfolio. I have 1.
It
Good
might
luck
be
next year in grade 11,
I
Angel.
just kept it.
Oh.
Ow! Ow! You were strapped to do it.
No, give it to us. Start handing things in.
Tony, we don't care.
No, I put it there, like the last term there.
No,
He said,
no.
put it there, put it there. Not there.
Probably in the back room. We don't care.
No, listen.
Those, no, listen to me. Those red things, listen. Next year, they're going to I've got your marks. I've got everything I need.
I only have 2 textbooks and 5 now.
Okay, Tony, did you show her that you had 4, Jay?
Ma'am, she went through my
Get
file.
out!
She went through my
Get
file.
out! Tony, put your portfolio on it. Tony, I'm going to throw something at you.
Ma'am, you're not going to hear the end of this.
No.
I sort of realised this.
Tony just put the file in.
Tony, go and play in the train.
I'm kidding.
Okay.
I do that on weekends, man.
Was it Tuesday?
Okay, so there we go. Currently, a site-wide mark book embargo in effect. Results for assessments dated from Friday 19th of June will only be available for viewing on the parent and pupil portals at the soonest on Friday the 31st of July
So by the time that we've written it, we can go
23:45. You may specify a later time and date if you wish.
So now can you not just...
So you'll
can
see
you just show us?
that it doesn't matter what date I say here for the release. 31st of July is. Why would somebody put an embargo on assessment marks?
What does embargo mean? Like a...
Somebody somewhere in the world must care about this. Whose life does it change if you get your marks a little bit sooner?
The school, because the school's very emotional. Everything's an issue.
You know, read our marks.
Okay, can I read them out loud?
No.
Okay, let's go.
No.
This is not a good
Isabella said I may.
Well, that's only Isabella.
Isabella said I may.
Who's Isabella?
Can we come to you and see our marks?
No,
It's just
that's Lucy.
It's literally
You kids need to learn to toughen up.
And I'm not seeing my marks.
It's a stupid test. No offense, man.
It's a
Stupid
test
test,
that
no
comes
offense.
off.
No, I don't take offense at that. What the hell?
Is this the substantive
Okay.
test, right?
Yeah.
I think that's what they're referring to.
Well, read the marks. No,
I would love to, but I was told no.
You told us to toughen up, so here we are toughening up. Read them.
That's you. Not everybody.
Not we.
Okay, man, can you excuse me, guys? Can you go to your communist party here?
Like, we just understand. We
I'm
just
just
got
the
an
name.
A.
Come on. It's an all or nothing. Either I read them or I
Okay, let's just suck it up now.
Can we sum it up, man? Go.
No.
J-Child, don't go down.
I've moved on. Okay, I'm going. So guys, 2 videos of brain dissections. Okay.
Hello, my name is Erin, and today we're coming to Live Lab to investigate the brain. This is a pig's brain, so this is going to help us to learn a little bit more about our own brain. So one of the most recognisable things we see about the brain are these little kind of grooves and
Did you do
bumps
this?
that we have. So the little grooves, these are called sulcus, and the bumps are called the
So
gyri.
there are 2 or
And
3
this helps to
terms that
increase
you
the
guys
surface
don't
area.
have to know.
Pig's brain doesn't have quite as many grooves and bumps as our brain would do. In fact, for our brain, about 2/3 of this surface is hidden inside those grooves. This gives us the biggest surface area, so it allows us to have more neurons, more brain cells. Okay, so here we can see the brainstem. So this is where all of our nerve signals are coming into our brain from all around our body. And as we go up here, we come to what's called the cerebellum, which literally means the small brain, the little brain. And this is responsible for things that need sort of very precision timing. And then we come up to the main part of the brain itself. We have the cerebral cortex there across the whole of this. And this is called the cerebrum. If we look just over here, you can see this little bit of black stuff there. The arachnoid mater is part of the meninges, which is a set of coverings that protect our brain. It's actually meninges that get inflamed with the illness meningitis. Our brains are split into 2 halves or hemispheres. So we have the left-hand one and the right-hand one. And actually your left-hand hemisphere of your brain controls the right-hand side of your body, and the right-hand side controls the left-hand side of your body. There is bits in between them though, so that the 2 hemispheres can communicate. This is called the corpus callosum. So let's have a look at our pig brain and see if we can find that. So this one's already been cut in half. The corpus callosum is this little structure that we can see in here. That's connecting the 2 hemispheres of our brain so that they can communicate. You might have heard of grey matter and white matter in your brains, so I can show you what that is. The white matter is this, it's actually kind of light pinkish stuff here, and the grey matter is the darker pink bits around the edge. And these are actually different parts of our neurons, our brain cells. So we have the cell bodies, and these make up the grey matter around the edge. And then our neurons have a long, thin bit that's called the axon that has a layer of fat all the way around. And those come in and give us our white matter. It's actually that fat that gives it this light pink colour. That fat's helping the messages to go through our neurons and helping to protect them. So when it comes to brains, does size matter? Well, if you look at our pig's brain, this is quite small. It fits easily in the palm of my hand. The largest brain in the animal kingdom belongs to the sperm whale. My brain is probably about the size of my 2 fists together like this. So not even close to the size of the sperm whale's brain. And I thought that I was quite smart. I've got 86 billion neurons in there. So perhaps it's brain size compared to body size that matters. As animals get larger, in general, they have larger brains. But humans have the largest brain size to body size ratio of any animal. If you'd like to see more insights on the outside, check out our heart and eyeball dissections. Coming soon, we'll be dissecting an egg in our hatchery. So for more science every week, click subscribe. Thanks for watching.
I'm so curious.
I desperately want you guys to do eye dissections. I just don't know whether,
Isn't it if you poke it, it like juices out?
Yeah, I'm still know.
no, it's a lovely dissection. Eyeball dissection at my best.
I want to dissect the heart. That would be cool.
Did you not dissect a heart last year?
Man, we literally, our grade is literally like the lab rats. We never do anything other grades have done.
Yeah, we just get tested with every single unit.
Yeah.
Okay, right, we go. Really steady. Go.
We're going to go inside and do some dissections of the brain and look at some of the important structures that are useful for understanding neurological principles and localization.
She's so cute.
Keep out.
The purpose of neuroanatomy and all of its tortuous tracks is to allow you to do neurological localization. That is to say, decide where in the brain of your patient the disease process is occurring. That's the total reason for most of you to want to understand the connectivity of the brain. Now let's look at the surface of the brain. We've removed the coverings, the leptomeninges, the pia, and the arachnoid, and I want to just show you the different lobes of the brain as an introduction. In addition, on this brain, you can see the brainstem. The brainstem extends from this region where my probe is, a little bit difficult to see, down all the way here to where the spinal cord was attached. In addition, you can see the cerebellum involved with coordinated motor control, as well as the surface of the cerebral cortex. This is the ventral surface of the brain. And the most prominent feature here is the temporal lobe, which I am outlining. Here is the temporal lobe on the other side. And in front of it is the undersurface of the frontal lobe. Cranial nerves are attached, such as the visual optic nerve and the sensory nerve from the face, the trigeminal nerve, and blood vessels, such as this large basilar artery, are running on the surface. Or you can even see the large internal carotid artery. We will come back and look at the nerves and the arteries and the other connections in greater detail. But I just want to introduce you to the region of the medulla, the pons, and the midbrain. And together they make up the brain stem. Let's look at the surface now of the brain and look at the different lobes. You already saw the inferior surface of the temporal lobe coming back to here, and we just kind of arbitrarily draw a line and say that this is approximately the end of the temporal lobe posteriorly and the tip of the temporal lobe anteriorly. And then we look for a sulcus on the surface of the brain. Sulci are these grooves, and the surface structures that are coiled are the gyri. So we have sulci and we have gyri. The gyri have names, but we're not going to learn most of them. However, this major sulcus, the central sulcus, because it runs from the midline all the way down to the lateral fissure, separating it from the temporal lobe, this central sulcus separates frontal lobe in front of it from parietal lobe behind it. And very interestingly, the cortex involved in voluntary movement is in front of the central sulcus, and the cortex involved with primary somatic sensation from your body is the gyrus behind the central sulcus. So this is frontal lobe, this is parietal lobe, this is motor cortex in general, and this is sensory cortex in general. So we're just getting our bearings. The frontal lobe is further sort of divided, and it's hard to see the distinction, but this band along here is the superior frontal gyrus. Then we have the middle frontal gyrus and the inferior frontal gyrus. The motor cortex in front of this precentral gyrus, as we call it, because it's in front of the central sulcus, is an area involved in the left hemisphere, which we're looking at, with the production of speech. And you may have heard of it before. It's called Broca's area. The rest of the frontal lobe is either motor function, planning motor function, or executive function, such as judgment and planning. Let's look at the parietal lobe now. The parietal lobe is separated from the occipital lobe by an arbitrary line that we draw between the parietal and the occipital regions, these little notches here. So if I were to display here, I would say that approximately this is the occipital lobe. The occipital lobe is very important in humans because this lobe is involved with visual information processing. It's a wonderful brain that we have, and we are very specialized as humans for visual computation or orientation and analysis. So we have our three lobes. Occipital lobe involved with vision, parietal lobe involved with sensation and integration of multiple modes of sensory input, and a frontal lobe with motor and higher order function. And this temporal lobe that is down here is very much involved with auditory information, which is coming into this fissure here that I'm pulling down. This is the lateral fissure. And auditory information, sound, comes to both hemispheres and is processed here. And this area behind it, the superior temporal gyrus, this back part here is involved in the comprehension of language. So you've got two language areas, an interpretive language area and a production language area. And when we study the consequences of cerebral stroke, we'll see that some strokes can knock out one area and you'll have problems producing intelligible speech, while other areas leave you hearing perfectly fine but not understanding what you're hearing, and other lesions or damaging strokes that involve all of this area can leave you with a large deficit, which we call global aphasia. Aphasia is a language disturbance. The temporal lobe, in addition to having an auditory function, has an area underneath this gyrus here, which is required in one